Mutations. Let's talk about them. So first of all, what is a mutation? A mutation is just any change to DNA or RNA. Anything that's going to affect that sequence of bases, those A's, T's, C's, G's, or U's, we call that a mutation. Doesn't matter how big, doesn't matter how small. Now, mutations can cause the amino acid sequence of a protein that a gene codes for to change, but they also cannot. We'll look at some examples of those. But if the amino acid sequence does change, this could result in the protein that gene codes for, right, that protein that's produced, not working anymore. Could also lead to the development of a new protein, and this is really important for new genetic information, for the generation of possibly new traits. And because of that, mutations are necessary for evolution to occur. Mutations are the source of genetic diversity, essentially. Mutations allow new traits to develop, and then organisms which have those traits might be better or worse suited for their environment. Those traits that are better suited for an environment, we consider those to be adaptations. And those that are worse, we consider those to be maladaptive. And so then natural selection and other evolutionary forces can select for those different traits, and we get eventually adaptation and evolution. So mutation, really important for that process. All right, we're going to talk about a few different types of mutations. First one is point mutations. And point mutation is when we just change one single nucleotide base in a gene sequence. So for example, if we have thymine in DNA and we replace that with a cytosine, that would be an example of a point mutation, also known as a substitution mutation, right? So right here, we see we've got a thymine here in this original sequence. If this somehow gets changed to a cytosine, that's an example of a point or substitution mutation. And you'll see either of those terms, point or substitution, they mean the same thing. We're changing just a single point, or if they're just like a substitute teacher, right? Comes in, replaces. And so that's what they are. But then we also have specific terms to categorize different types of point mutations based on how they affect the protein. And so you can see those down here. So the first one is called a silent mutation. And a silent mutation is where you can change the DNA or the RNA, but you don't actually change the protein. And this is the case, or this can happen because the genetic code is redundant. And by redundant, we just mean that there are multiple codons, multiple little sequences of 3 nucleotide bases that code for the same amino acid. If you look at your codon table, you'll see that over the place. There might be 3 codons that code for this one, 4 that code for this one. There are even some where there's like 6 codons that code for the same amino acid. Right, so for example, CAA in mRNA, if you look at your codon table, you'll see that gives you glutamine. If we change CAA to CAG, that now codes for glutamine. So it's the same thing. There's no effect on the protein. And so this is a way where DNA or RNA can change, but it doesn't really infect the organism at all. The protein that's produced by that gene is exactly the same as it was. There's no functional difference, no structural difference. And so we call that a silent mutation because it's like you can't hear the effect of the mutation. Next up are missense mutations. Missense mutations are where you actually do change the amino acid to a different one. And so if you take CAA again and we change it to CCA, that gives us proline. And proline is a different amino acid from glutamine, and it's going to have different chemical properties. And so it's possible that having a proline instead of glutamine is going to change the structure of that protein in some way. Now, the effect of this could be variable. It could make the protein work a little bit differently. It might not have much of an effect at all. It might make the protein work a little worse than it did before. It might make the protein actually work a little better than it did before. It's hard to say. It just depends on the protein and its structure and the way that the mutation affects that, right? There's no real way to know that except we're like analyzing every single mutation. Then we also have nonsense point mutations. A nonsense point mutation is where you actually take a codon and you change it to a stop codon. So if CAA in the mRNA becomes UAA, that's now a stop, and that's going to cut off protein synthesis, going to cut off that translation process in the middle of the gene. And this is usually going to have a serious effect. So this is usually going to cause whatever that protein is that was being coded for that gene to no longer work. And so depending on what that protein is and what it does and how important it is and what kind of impact it has on the organism, it could be really serious. It could be a deadly mutation. It could be a mutation that happens to a gene that's so critical for development that an organism that has this mutation doesn't even wind up developing and doesn't even make it to birth, for example, if you're thinking about like humans. This could wind up resulting in a miscarriage, for example. But it also could just mean that, the organism that has that can't make a particular pigment molecule, and so they're an albino. It could be that, right? It just depends on what the gene is, of course. All right, then we got frameshift mutations. Now, frameshift mutations are a little different than point and substitution mutations. So this is like our second major class of mutations. And these are mutations that change the reading frame of the codons in the base sequence. And there are two ways to do this. We can add extra nucleotides, which is called insertion, or we can remove nucleotides, which is called a deletion. And what happens is when you add or remove codons, it shifts the way that the ribosome reads the mRNA. Because remember, your ribosome reads mRNA 3 letters at a time, right? 3 bases at a time. That's just how it works. Once it reads that start codon, that establishes the reading frame. It sees AUG. All right, we start here. Now we're reading every 3 letters, no matter what they are. And the ribosome just does its job according to the way that the chemistry of the ribosome and the amino acids and everything works. All right. So if you change the reading frame, you potentially change every codon after that mutation. So to help understand this, we're going to use an analogy here. Imagine we have a sentence, right? Because genes are kind of like genetic sentences, right? Or genetic paragraphs, depending on how long they are, that are all made of 3-letter words, right? Codons are 3 letters. And so here's a sentence with all 3-letter words. The big cat ate the fat rat. All right. Now, if you're typing this up, and you make a typo, somebody else reading your sentence is probably going to be able to figure out what you were meaning, especially because we have spaces between the words, right? Well, there are no spaces between codons. There are no spaces in the genetic language. And the other thing is that unlike your friend who's reading your sentence who can spot your typo, a ribosome can't think. Ribosomes just are doing chemistry, right? So ribosome is going to read 3 letters at a time no matter what. And so if we insert a T in the middle of this, the ribosome is just going to keep reading 3 letters at a time. The bit gk tat f f tra ta. Bunch of nonsense, right? We can see that every word after the has changed. Likewise, every codon after an insertion frameshift mutation will change as well and will potentially code for a different amino acid. Same thing if we delete, right? So we take out the A here from cat, and we get the big tet hef atarat. Once again, nonsense. Everything after the mutation has changed to something else. We still have the big, and likewise with frameshift mutations, any codons that have already been translated before the mutation, they're going to be the same. It's only everything afterward, right? Because we are shifting the reading frame, shifted over by a letter or 2 letters. Now, if you have a deletion or an insertion of 3 nucleotides, you actually don't shift the reading frame. You would actually just insert or delete an entire amino acid, which obviously could have impacts as well. But anything that's a multiple of 3 mutation with an insertion or deletion wouldn't actually shift the reading frame, but 1 or 2 or any of those are. All right. So codons in DNA and RNA are changed in the exact same way. All of those codons after the mutation are going to be different. So here's an example. Here we have a protein here that's reading lysine, threonine, alanine. If we add an adenine here in between A, C, C, then we actually wind up with different amino acids, right? These are different than these ones. And eventually, if you keep going down, eventually you'll reach a stop codon a little early too. So it's kind of like a 2-for-1. You shift the reading frame, you change all the codons, you change a bunch of amino acids, and then you'll often get an early stop codon as well. So it's kind of like it's a nonsense mutation too. And so deletion, same idea. We remove this thymine here, and we can see that was serine, leucine, valine, is now histidine, serine, tyrosine. These are different. And later on, 38 codons later, we once again have an early stop codon. So frameshift mutations are generally going to have a much bigger impact on the structure of a protein and therefore the function of the organism than point or substitution mutations. Point substitution mutations can still have big impacts, especially if they are a nonsense mutation, right? Nonsense mutations are going to have a similar level of effect as a frameshift, one of those insertions or deletions. But, with a point or substitution, you can also have a silent mutation or a missense mutation, which generally don't have as big of an impact. But some missense mutations do. So some mutations, they can be beneficial. Other ones can be harmful. Beneficial mutations, like we talked about already, help organisms survive and can potentially help them survive and reproduce, right? We call these adaptations, right? These are important for evolution to occur. Harmful mutations can result in disease or death, can cause proteins to not function right or not function at all. And if they're really important proteins for something, then that can lead to a lot of problems. Harmful mutations can cause genetic disorders. They can lead to cancer, lead to all sorts of nasty stuff. All right, now, here is an example of a missense mutation, right, where we just swap one out that actually has a pretty big impact. So this is how sickle cell disease works. So in sickle cell disease, we actually have a point mutation or point/substitution where we just swap one letter that causes one amino acid to change. So it's a missense point mutation, but that one amino acid makes a big difference. So hemoglobin, that protein that carries oxygen around in your red blood cells that your red blood cells are full of, right? That hemoglobin protein normally has this sequence here, but in this sequence, if you change this glutamic acid to a valine, valine has a very different chemical property from glutamic acid. Valine is hydrophobic, whereas glutamic acid is hydrophilic. And because of that, when you have this valine here, it allows for parts of the hemoglobin protein on this beta chain to become attracted to a hydrophobic pocket on a neighboring hemoglobin. And so they wind up connecting to each other and forming these big long strands, these big long fibers. And those fibers are what stretch out the cell and cause the cells to sickle, and leads to all the different problems that O.C. Ryan had when we learned about her during presentations. So that's an example of a disease that's caused by just one DNA base being wrong. Oops. Wrong way. There we go. Similarly, NF1, right, neurofibromatosis type 1, the mutation, the genetic disease that Kai Rossi has, is a mutation of a particular gene. Now, there's a variety of different ways that this particular gene can become non-functional, right? It could be a nonsense mutation. It could be a frameshift mutation. It's usually some type of frameshift, an insertion or deletion that happens. But neurofibromin is a protein that's produced by the NF1 gene. And if that neurofibromin protein isn't produced or isn't functional, then you wind up having tumors come up. So neurofibromin, the protein, acts as a tumor suppressor. And the reason it's called that is because it's named after the fibroma tumors. It's kind of weird the way that things are named often. So basically, scientists observe these tumors. They called them neurofibromas because they're like a fibroma tumor. They're this non-cancerous, fibrous little bump, basically. And they called neurofibromin because they generally grow along nerves. And so that's where that name comes from. And so then they eventually figured out, oh, hey, this protein is important because if this protein works, you don't have the tumors. If this protein doesn't work, we do cause a tumor. So then they name the protein after the condition. And so it's called neurofibromin, but it actually prevents neurofibromas. I know, kind of weird, right? Anyway, that protein acts as a tumor suppressor, prevents tumors. So if that protein doesn't work, if somebody doesn't have functional neurofibromin NF1 genes that produce that protein, then they wind up developing these tumors. So mutations in this particular gene here on chromosome 17, right, could be frameshift mutations, could be nonsense mutations, but something that's going to cause that NF1 gene to no longer be produced or no longer function leads to this condition where you have these little tumors develop called neurofibromas. So it's because that neurofibromin protein isn't being produced by that person's cells. All right, we're going to talk more about NF1 and the NF1 mutation and how it is inherited in the next activity.